Okay, so welcome back to the second section of photosynthesis for Bio 348. We've spoken about photosynthesis, we've spoken about the light reaction, we've spoken about the Kelvin-Benson cycle. We started looking at how the environment can play a role in these factors and influence them, and we're going to continue now. Um, Before we get into the nitty gritty, we need to understand uh, this concept of light. Remember, we were looking at all those things that could influence photosynthesis. We mentioned light, atmospheric CO2, temperature, humidity, etc., and abiotic stresses. So we want to look at light. And in order for us to do that, we need to understand what it means to quantify usable light. And what I mean by that is not all the light that comes to the plant via the sun is used, and also specific wavelengths are used. So when we say something is a flux, or in a lot of measurements when it comes to photosynthetically active radiation and etc., 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 a lot of the words and terminology we use is flux. Um, and this is just energy, light energy that is being measured. The measure of this light energy, or photons. So if I say photons, it's light energy. If I say irradiance, photons, flux, same thing. Um, Photon irradiance, it's just different ways of measuring photons, measuring that light. So measuring the way the light reaches the leaf, and how we would quantify it. So don't get bogged down by these equations thinking that you have to know them. You just have to understand that we need to be able to quantify usable light. We need to know what is hitting the leaf, what is being absorbed and being used in photosynthesis. In order to do this, we need to be able to quantify it. So we use terms like irradiance and photoirradiance, or photon irradiance, and they're just different ways of measuring usable light. Okay, so it's easier for us to explain what's happening. Everybody should remember that photosynthetically active radiation, PAR, is between the wavelengths of 400 to 700 nanometers. Remember, 400 will be that blue zone, that blue purple zone, and 700 will be that red orange zone. Remember, this 400 nanometers has more energy, and the 700 nanometers is lower in energy. Remember, I showed you, we looked at photosystem 1 and photosystem 2. We said photosystem 2 is 680, whereas photosystem 1 is 700. That is the nanometers that it absorbs at. And when we talk about photosynthetically active radiation, we would rather refer to it as quanta. Now, this is things that when you start measuring, you'll have to start paying attention to. Um, it isn't something that I would really, freak out about and stress to get in my mind. It's just you need to understand these terms. So when we're talking about photon irradiance, when we're talking about quanta, when we're talking about quantum yield, and we talking about all of that, we're referring to that usable light and how we would quantify it, how we would make it a number for us as plant physiologists to be able to understand what's happening to the photosynthesis. So here is the 400 and the 700. Here's that visible spectrum. This is the blue, the purpley blue, and there's this red orange, and this is where the absorption of chlorophyll is. So the reason why it's don't get confused. The reason why the plant is green is because it will be at that radiation, at that wavelength, it reflects back. So we see green because it doesn't absorb the green light, it reflects the green light, it absorbs the purple and blues and the yellows and the orange and the reds. Reflects the green, absorbs the others on the other side of it. And you can see this dip over here where it's absorbing, it's not absorbing all of it, and this will be where the reflecting of green is taking place. So just have a look at this for a minute and see how much light the sun is actually giving off. It's giving off an incredible amount. Not all of that is used. Any wavelength but lower than 400 and above 800 is going to damage the photosystems. It's going to damage the proteins. It's going to damage specifically that D1 protein in photosystem 2, and it's going to cause photoimbibition. We'll get to that. But what you need to recognize from the slide is that the sun is producing a lot of energy. Not all that energy gets absorbed for photosynthesis. A lot of it gets reflected, and we will see later. So the incoming light can strike a flat leaf at various angles. Remember, we spoke about the orientation of that angle. Remember, we spoke that depending on how the leaf is orientated will determine how much light it absorbs, and as a result, what is usable and what is not usable. So if you look at page 245 on in your recommended textbook, which is Tays and Zager, you will see a little diagram that explains what I'm about to tell you. And what it is this point over here. When it is not perpendicular to the leaf. So watch my mouse. If there's a 90-degree angle, it's perpendicular. If it is not perpendicular or it deviates from that perpendicular incoming light, we have to recognize that that energy is going to be lost and it's not going to be absorbed by the leaf. And so we have to take into consideration at what angle it's hitting. And because we have to do that, we will minus that cosine. So it's a little equation. By all means, that's not going to make or break this module, but you do need to recognize that the orientation of the leaf is going to determine how much light is absorbed, how much light is used, etc., etc. And if it deviates from a perpendicular angle, we have to use cosine, cosine alpha or cosine of the angle to try and sort out that the quantifiableness of it. So just from that second point, have a look at the picture if you want to. All you have to know is that the angle at which it hits the leaf will determine how much gets absorbed, how much usable light gets used, and it's a way of quantifying this so we know how much is lost when it is at different angles. So on a sunny day, your photosynthetically active radiation is about 2,000 micromoles per squared per meter squared per second. And this is a nice sunny day. This is what's on average, can be higher, but that's around average for most plants. Whereas now, what this point is saying is that that's in full sun. Plants are not always exposed to that high amount of photosynthetically active radiation. Sometimes there will be a canopy, and up at the top will be 2,000, and down at the bottom of the canopy of a great big forest, there'll only be 10 micromoles of photosynthetically active radiation. And this plays a big role. This plays on a big role what plants can be grown there. It plays a role as to how the photosynthesis is going to adapt to deal with this low light. Because remember, we're looking at what affects photosynthesis. We're looking at light, and if we've got a low light situation, how will the photosynthesis apparatus adapt to handle the low light? Okay, so remember, the leaf anatomy maximizes light absorption. So remember what we said, not all the energy that comes from the sun that reaches the earth is used in photosynthesis. In fact, less than 5% is used in photosynthesis. Remember that we've got a specific wavelengths that get absorbed, photosynthetically active radiation, which is between 400 and 600. And then we've got a little gap in here where the green band is that reflects it back. So of the light that is absorbed, about 15% is reflected or transmitted, and that's what gives off the green light. Now, your chlorophyll, remember we said photosystem 2 absorbs at about 680 and photosystem 1 at 700. Now, remember 400 nanometers has more energy than 700 nanometers. So there's more energy in these wavelengths than there is here. So the reason why the chlorophyll pigments, the specific chlorophyll reaction center, that special chlorophyll A in photosystem 2 and photosystem 1 tend to be on the red-orange side of it, is because that is weaker energy. And the reason for this is that 400 is that purple-blue area has a lot of energy, and it could damage the proteins. It could make serious damage to that D1 protein, and that D1 protein is what makes up photosystem 1 and makes up photosystem 2. And if that is damaged, we get photoinhibition, inhibition, photoinhibition. Inhibition is when you put seeds on a shaker in distilled water and it imbibes. Sorry. Photoinhibition. So the chlorophyll needs to be protected. And so we have other pigments, accessory pigments that absorb that 400, that bluer side, to prevent damage to the photosystems. Of the 85% of photosynthetically active radiation that's absorbed, so remember we've spoken, so what of the photosynthetically active, so not all the energy that comes to the sun, less than 5% is photosynthetically active radiation, and of that 15% is reflected or transmitted as green, and Of that 85, a massive amount is lost to heat, to chlorophyll fluorescence, and only a small bit is actually absorbed. Remember I said that orangey yellow area is what gets absorbed. All that other is transmitted back as chlorophyll fluorescence and is dismissed or uh, given off as heat energy. And this is all to protect those valuable photosystems. So this is just a visual picture of what comes, so of what actually comes from the sun. Non-absorbed wavelengths, obviously those wavelengths that are UV and above, so only between 400 and 700, that's about 40% of that we get reflection and transmission and heat, and then metabolism, and then carbohydrates. So only 5% of this energy that comes from the sun is actually what is used in photosynthesis to give us the carbohydrates that will give the plant the carbohydrates that it needs. So the plant needs to have protection mechanisms in place to handle and withstand the intense energy that comes from the sun, which it does. It has accessory pigments, carotenoids, xanthophylls, etc., etc. Okay, so the anatomy of the leaf, which we've discussed, is highly specialized for light absorption because its primary job is photosynthesis, and photosynthesis requires light. Therefore, the leaf must be able to absorb light. It must also be able to capture enough light because not everywhere is homogeneous. We have areas where not necessarily the plants are going to get 2000 micromoles per meter squared per second. So not all the plants are going to have access to that amount of photosynthetically active radiation, and because of that, the plant needs to adapt its leaf to make sure for optimal absorption. And for that, it has a transparent epidermis. Its cells are often convex and convex. So think about glasses. I wear glasses, and without them, I'm as blind as a bat. But when you look at concave and convex, remember convex focuses the light, concave will refract the light back, it'll send it back up. But convex, so if you reaching out to capture water in your hands and you put them together so that the water can hold it, the way you've got your hand in like a little bowl shape, that's convex. And we've got these different epidermal layers which focuses the light to these chlorophyll. So we've got this upper epidermis, that waxy cuticle, it's transparent so that the chlorophyll can clearly be seen, the chloroplasts can clearly be seen. We've got these multiple layers, and this is dual fold. This idea of the sieve effect and the light channeling is dual fold. Um, the sieve effect is this idea of the light is going to come through and it's going to bounce and it's going to bounce and it's going to bounce, but there's so many layers that it will eventually be absorbed. And this light channeling is that there's various layers, and if what comes in here, if it slips through, it'll get absorbed here. If it slips through, it'll get absorbed here. If it slips through, it'll get absorbed. So this light channeling that it will eventually get absorbed. Now, chloroplasts, the volume of chloroplasts between surface to ratio is dependent. If you are in a very shady area, you're going to increase your chloroplasts, because an increase in chloroplasts means you have a higher surface to volume ratio, which will increase efficiency. If you're in a very sunny area and you're getting that 2000 photosynthetically active radiation, then you're going to decrease your chloroplasts because you've got enough sun coming. So the plant plays with that surface to volume ratio to increase efficiency. But it also it's a problem, because if so, this last point asking, are multiple layers really efficient? If you're in the shade, perhaps not, because not everything is going to go down to the bottom. So you don't really need multiple layers. Whereas for something like something that's in the sun, it's a good idea. So the leaf is adapting to harness the harness light most efficiently, depending on its environment or environmental situation. So this is just another example. There we've got um, our photosynthetically active radiation. Remember, this near infrared does not get absorbed, it gets reflected. And we've got this spongy mesophyll that allows for the scattering kind of effect. Now, remember I said that the amount of energy that actually comes from the sun is enormous. And what the plant actually absorbs is a lot less. It's less than 60%, much less than 60%. 60% is non-usable, it's non-absorbed. That doesn't mean that the plant doesn't get hit by it. So the plant needs to have contingencies in place to protect itself from excess light. This is where we start getting hairs and salt glands and cuticle waxes and fine hairs on the leaves to dissipate, etc., etc. So, and also high xanthophyll, high secondary pigments such as carotenoids and xanthophyll, etc. So the plant needs to be able to adapt its morphology to ensure that it can maximize absorption of photosynthetically active radiation, and at the same time the plant or the leaf needs to adapt to be able to protect from excess light energy or excess light. So finding that delicate balance is really special, and now you've also got to remember that the plant is sessile. So if the environment changes, the plant needs to be able to adjust that. And this is where we get this idea of plasticity in. If the plant can still change within reason, it is known as phenotypic plasticity or plasticity. And it's the plant has to be able to adapt to the specific set of scenarios that the environment is giving it. And you have to be able to identify by looking at a plant, okay, yes, this plant was in sun or because of this, and this is a modification because of this. So we're looking at light. So if we have a look here, you can see this beaver tail cactus. So everybody knows that uh, cactus is singular, cacti is plural. That cacti grow in very hot desert conditions, and they have a very thick stem. And if I were to say to you now, as a Bio 348 student, what has this plant done to protect its photosynthesis? You need to be able to go, okay, well, first of all, that red over there is definitely signs of xanthophyll carotenoids, which is there to absorb and reflect excessive heat. It's got thick leaves, and that thick leaf is to store water. The photosynthesis is going to occur at night, so that during the day it's going to collect the energy, the light reaction is going to happen, it's going to store the carbon dioxide, and during the night it's going to close its stomata, so to prevent excess sorry, during the day the stomata is closed, and during the night the stomata is open, where we can get gaseous exchange to prevent excess light. If we have a look at this cactus over here, you can see this kind of fuzzy white stuff all over here. Those are actually hairs, and those hairs are twofold. Number one, it provides some sort of shade by deflecting the light back. So it captures the light before it reaches the mesosphere and reflects it back as excess heat, as well as it prevents excess water loss, because you're capturing an excess amount of light. Your stomata have to be open because it's going to be hot, which means you're going to lose water. And if you lose too much water, you're going to shrivel up and die. And this manages to keep the water trapped in that region so that it can reabsorb it. And this California fan palm, also very similar. It's not letting its leaves fall. It's not, senescence hasn't completely happened here. The leaves haven't fallen off. And the reason for this is because this provides a kind of like a safety net to keep the water within it, so that it keeps itself cool. So there's all sorts of stuff that plants have developed to ensure that excess light doesn't damage the photosystems. Hair, waxy cuticles, cam, photosynthetic rate, those are all examples of ways plants have adapted to handle excess light to prevent photo inhibition. Okay, now when you start getting to a forest kind of a vibe where you have multiple species, various growth forms of plants, I, I don't know if everybody got what I mean when I say growth forms, because I said it on the first prac and I wasn't quite sure. So, I am short, I'm plumpy, um, as I can eat as much vegetables as I like. I'm never going to grow taller. So this is my growth form. It's in my DNA. I'm not going to grow any taller. So it's the same thing. This maize growth form is that they're long, tall, taproot, fibrous roots, etc. Whereas the pea is a taproot, it's shorter, it's broader. So when I say growth form, it's just the DNA marked morphology of the plants. So when you start getting into regions where we have areas with multiple different growth forms of plants, you're gonna have um, plants competing for sunlight. And it becomes important because plants that are pioneer plants might not necessarily be mature adults that survive over the generations. So you'll notice if you take a walk through a forest and there's a gap in the canopy, you've got all these little new seedlings coming up, and it's a race to the top. And that's what we mean compete for sunlight. You do have plants competing for sunlight. Um, so it just obviously you need to remember now, photosynthesis gives us energy, photosynthesis gives the plants the energy they need to grow. So being in a shaded environment, you have a lower quantity of glucose being made and therefore a different quality and lower photosynthetic rates. Okay, so obviously your growth form, if you are higher and you have more branches and you can branch out, you increase your chances of light interception and ultimately light absorption. So this is what I meant by forest. You can see that these trees are nice and tall, and these guys that are at the bottom, they really have a brief period where they get sun. And what happens is these plants at the bottom of a canopy are so well adapted. We've got these moments of sunflex that come through the canopy, that break through the canopy onto the floor. So they've got this short, really short time of intense sunlight, and they eat it up. That those light reactions just fire, it gets all the energy in that brief short moment, but they're adapted to it. That's how they've always been. There are the plants that grow at the bottom of canopies because their genes, their genotypes are designed to capture this light in such a brief short moment. Okay, so we know that the light causes photolysis. Photolysis is the splitting. Sorry, I'm going back to the light reaction. We're going back to where in the light reaction the water splits to give us the oxygen and to give us the hydrogen plus ions, so that photosystem 2 can take the electrons from the hydrogen plus ions so that they become hydrogen plus ions. We know that light is necessary for this process. We do not know how the light splits the water. We don't know if the light is doing the splitting of the water, so breaking up the molecules to break up the oxygen and the hydrogen, or if light is the catalase to splitting the water. Not sure, and this is just explaining that even though we know so much, there's still a lot that we don't know. And one of the major questions that perhaps one day one of you will answer is, we're not sure what causes the water to split. Is it the light, or is the light catalyzing something that causes the water to split? And then the photosystem 2 to take those two electrons from those two hydrogens, leaving us with the two hydrogen plus ions and the oxygen that binds to give us oxygen 2. We also know that this is a very self-sufficient renewable process. It's a renewable cycle that can keep producing on its own, which we know. Photosynthesis, but have you stopped to consider that take humans out of the world today and plants will be fine. In fact, they'll survive just fine. This is a self-sufficient. I mean, if I want to eat something, I have to make a sandwich using external forces. I can't make the sandwich myself. My body can't make my own sandwich. I have to make the sandwich myself. The plant's making its own sandwich. That's amazing. It's self-sustaining, it's renewable, and it's paving the way for solar power research because it is such, it's just insane. This process of photosynthesis, making your own energy from light energy to chemical energy, it's mind-blowing and we take it for granted. Okay, so remember the leaf orientation or the leaf angle will affect the light that is absorbed, and obviously if you are at steeper angles you will receive less sun during the day or not. Okay, so so just think about this for a moment. If you are perpendicular to the light that's coming, remember I told you about page 245. When the light hits that leaf and the sun moves, it doesn't stay still. Well, woo, flat earthers. The movement moves. The earth moves. The sun stays still. We go around the sun. But anyway, when when the sun is at noon, you've got the sun hitting that earth, the leaf fully. And as we go through the day from morning till evening, the leaf is getting as much light as possible. If you have a steeper angle and you are straight up, you're going to get it for either just the morning or the noon. That afternoon phase is going to hit you. It's going to be vertical. It's going to be useless. So when we say, why are the leaves at the top of a canopy having steeper angles? Remember, at the top of a canopy, at the top of the forests, you've got almost 2,000 photosynthetically active radiation, micromoles. And you've got to protect your photosystem. You've got to make sure that the excess light doesn't damage your photosystem. So having a steeper angle will condense the amount of time that you have light access for, whereas the angle decreases as you go lower because obviously it needs to capture as much as it can throughout the day. And then you specifically get plants that do solar tracking. A very popular plant that solar tracks is the sunflower, where it will actually follow the sun throughout the day and go from one side to the other side as it collects sunlight. So a pulvinus, this is a pulvinus over here, and this is what's going to change the angle of the leaves. It's going to change how the leaf moves, whether it's an angle or low angle, and it's all controlled by water concentration. So the cells in there are controlled with water concentration, and as it becomes turgid or it decreases its water within the cell and becomes more flaccid, the angles will change. It's different for every plant. You can see here this pulvinus looks different to this one, but this is ultimately, that's going to determine how steep the angle is or not, and or how it's going to move during the day, solar tracking. So that's the job of a pulvinus. So then remember tropism. Tropism is to move towards. So heliotropism is towards something. And obviously helio will be the sun, whereas phototropism will be specifically towards light, whereas helio will be specific towards the sun. You also get gravitropism, hydrotropism, etc. Not to be confused with nastic movement. Be careful, tropism is towards, nastic movement is just movement because it was stimulated. So heliotropic to bend towards the sun, dioheliotropic to actually follow the sun by solar tracking, and paraheliotropic to avoid. Make your leaf straight up to avoid excess light. Make your cycle short-lived. Make sure you've got, short life cycle. Prevent water loss. Okay, so as we're discussing light, we need to understand, remember I've mentioned plasticity. So remember I told you in the first set of slides, if I'm cold, I put on a jersey. If I'm hot, I go for a swim. A plant can't do that. A plant can't put a jersey on, and a plant can't go for a swim, and it can't get up and move. So it has to be able to adapt. And the plant being able to adjust its phenotype is known as plasticity. Now, the genotype, the genes, controls the phenotype. But being able to manipulate and change your phenotype within the limits that the genotype gives. So, for example, just because I am a pea plant does not mean now through plasticity I can become a maize plant. It's not going to happen, or as tall as a maize plant. I have certain genotypic limitations that I cannot alter, but I can alter my phenotype within my genotype's limits, within that threshold. Now this, we can see the plasticity between the same species growing as sun plants or shade plants. And this idea of acclimation is just a process where the biochemical, morphological, and physiological, so your biochemical will be ROS, antioxidants, etc. Your physiology will be your photosynthetic rate, your chlorophyll content, etc., etc., where your morphology is how many chloroplasts, leaf area, etc. So now you can have, uh, so now I take a species, two plants, same species, I grow 1 inch sun, 1 inch shade, and they adapt. And they become specialized. And then I take them out and I switch places with them. And they are not able to adapt quick enough. So you can't just, this is a process that it changes as it grows up. As the plant grows, it adapts, it changes, it becomes what it needs to be in its region. If I uproot it and put it in a different region, does not mean it's going to survive. If it's within the if some plasticity is um, definitely reversible. If it is still reversible, it's plasticity. If it is specialized, that specific leaf or that specific plant is for sun and that specific plant is for shade, no go. So plasticity, we can still change back and forth. Our genotype allows us to change the morphology back and forth. If we are specialized for sun, doesn't matter if I put you in shade, you're not going to grow. If you specialize for shade, I put you in sun, you're not going to grow. Okay. So, this is the common nettle. Both plant A and plant B are the same species. They are both the common nettle. Picture A has been grown in sun. Picture B has been grown in shade. Take a minute, pause the video, and try and see what the plant has done to its morphology, how it's changed its morphology to adapt to the sun and to the shade. Okay. The shade plant leaves are larger. Remember, the bigger your leaf, the more chlorophyll content you have, the more chances you can absorb. Sun plants, smaller leaves. You need to be small because you're in full sun. You don't want to damage, so small, less surface area to get hit by too much sun. And so that there's also not a large surface area for loss of water. Shade plants have longer internodes. So internodes are between the leaves, this bit over here. They have longer internodes. They have longer internodes because they're looking for more light, whereas sun, shorter internodes because we've got sun. Why must I put effort into sun? Let me look for something else like water or nutrients. Sun plants will have thick cuticles to reduce the loss of water. Sun leaves are thick. They're thick so that we have that sieve and that channeling so that it gets lost as heat so that it doesn't damage the photosystem. Shade leaves are thinner so that they capture as much as possible. The chloroplasts are evenly distributed. The sun leaves often red in pigmentation, and this is due to anthocyanins, which is a type of red pigment. So xanthophyll, chloroplasts, carotenoids, it's just a pigment that will reflect that red wavelength. So to prevent excess absorption of energy that will otherwise have to be lost to heat or actually causing ultraviolet damage. Remember, the color may differ. I mean, these two, you would think they're not the same species. We don't need a lot of chlorophyll in this, which is why we've lost that rich greeny color, because we've got enough sun. Whereas here, we need as much chlorophyll as possible. So, like I said, more chlorophyll. Okay, so here's a moment. Shade leaves have more chlorophyll. Why? Because the light, remember, supply and demand. We want to photosynthesize. We need to absorb sunlight. So we need more chlorophyll so that we can get supply for our demand, whereas sun leaves have more rubisco because we've got light, we've got carbon dioxide. What's our limiting resource? Our limiting resource is rubisco. If we have more rubisco, we will be able to photosynthesize more. That Calvin cycle will be able to happen faster and we will get more glucose. Okay, so we get a point. So later on, so we're gonna see in the light response curves. But remember, I told you that photosynthesis, plants photosynthesize, but they also cellular respirate. So this last point here is saying that, remember, I said that plants photosynthesize and they respire, but the photosynthesis just it shadows, it overshadows the respiration. We don't see the damage or the effects of the respiration because the photosynthesis covers it and then earns a profit. So this last point that it says there's a point where the photosynthesis equals the rate of respiration. This is known as the compensation point. This is lower in shade plants. This means that in shade plants, they're not going to photosynthesize more. They photosynthesize less. As a result, the plants do not use their energy as much because they have to conserve it. They have to conserve it for what they deem to be important or whatever. And because they're not photosynthesizing as much, they're not respiring as much, which means they have a lower compensation point, which ultimately means that they can photosynthesize more efficiently at low levels, whereas in sun plants, the compensation point is higher. So, depending on how much light we get, if we have too much light or too little light, it can damage our growth, the plant's growth and reproduction. You need to understand that there is a relationship between the photosynthetic properties and the way and the environment around it and how the plant adapts to the environment around it. We need to have a look at light response curves, which we will, and this is just the photosynthetic responses to light, which is measured by light response curves. Put a star, put an exclamation point. Light response curves are so important. And these same curves can be used to compare sun and shade plants and also C3 and C4 plants. So we can use photosynthetic curves which compare the photosynthetic rates to light. We can compare sun and shade, and we can compare C3 and C4, which is what we're going to do. Okay, so why is the light compensation points lower for shade plants? Remember, the respiration rates are very low, and because the respiration rates is low, the photosynthesis, little photosynthesis is needed to bring that net. Remember, exchange zero, where the photosynthesis equals the respiration rates. Because of the low respiration rates, it allows the plant to survive in limited environments or limited light environments, which means they can photosynthesize at lower photosynthetically active rates. So it's kind of like when you're cold, when you're too cold or when you're too hot, you don't want to do anything. You want to conserve your energy. It's the same thing. They're conserving their energy, and because they're conserving their energy, they only need a little bit of photosynthesis to reach zero. So that's why the compensation point is so low. So, remember, we were talking about why we were seeing some red pigments in the stem and in the leaves. Well, we have different type of pigments that can readmit the light or transmit the light to avoid excess light energy from damaging the photosystems, and this is known as the xanthophyll cycle. There are three, it's a type of carotenoid, or you remember carotenoids, the orange pigments. So why are carrots orange? They're because of carotenoids, and if you tell me that carrots were a different color at one stage, I don't care. Well, they're orange now because of the carotenoids. 10 points to whoever can tell me the story about carrots in our next practical when you see me next. But anyway, so this cycle, there's three. I love you guys, but I'm not going to try and pronounce those names as much as I would like to. There's Biol one, the anther one, and the zea one. They are all carotenoids that are designed to readmit excess light energy to avoid the photosystems from being damaged. So this is an example. Remember what we were talking about with the sun and the solar, that we've got a lot of energy hitting the plants, and as a result we've got all this excess light that needs to be dissipated. And only this little bit is actually photosynthetic. So here's the cycle that we were talking about. This line is light, and remember, look at the times here. At 12 o'clock, um, it's going to be the hottest time of the day. I know for me, my mom always used to say, Candice, between 11 and 1, you're inside the house, otherwise you're going to get burnt to a crisp. So yes, that is the hottest time of the day, and you can see the cycle there of those three xanthophylls coming in and doing what they need to do. Oh, sorry, got lost. Okay, so remember, leaves that are grown in full sunlight are going to have a higher xanthophyll pool or a higher xanthophyll concentration, whereas opposed to those that are in the shade. But also, don't forget those plants that are exposed to sunflex. This is very important for them as well. The xanthophyll is found in all plants, just different content, just different amounts in each plant. Um, Chloroplasts also move. So I think you do a prac in Bio 101. I hope you guys did it, where you actually see the chloroplasts turn when you put light on. So it's very beautiful, very cute. Anyway, um, the chloroplasts can move within the cells, within the plant cells, and depending on how, remember, we were talking about the leaves being perpendicular, well, the chloroplasts as well. The chloroplasts, if they're perpendicular, meaning they're perpendicular to the light, they'll absorb more, but they do turn so that they're not all the way. Let me see. So here you can see it in the picture. You'll see they've gone to the edge of the walls and they've turned around so that they don't absorb too much light, so that light will just go straight through and not damage their photosystems. This is that strong blue light, remember, 400 nanometers. Okay, so remember I was telling you too much light will lead to photoinhibition. I called it inhibition, and remember that excess can damage photosystem 2, specifically that D1 protein that makes up photosystem 2. Um, Now, there's different types of photo inhibition. There can be dynamic photoinhibition, where it's moderate, but and the quantum efficiency decreases a little bit, but the photosynthetic rate remains unchanged. It's still going on. It's battling. It's going to take a bit longer, but it remains unchanged. And this can be caused because there's just too much light energy, so they have to focus on getting rid of the heat, open your stomata, transpiration. So it's not as fast, it's going to take longer. It's temporary, and everything can still recover good as normal. It got a cold and it's back normal. Um, so you get this photosynthetic process over here, absorbed light. Dynamic inhibition, we're reaching this point a lot later than we would have in optimal, whereas chronic photoinhibition with da, 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 na, 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 it's dead. No point of no return. That photosystem is damaged. It's not going to work anymore. It's retired. Just get rid of it. It's broken. The plant will break it down and reuse the nutrients for other stuff. So chronic, dead, dynamic, little bit of damage, a little bit slower, but we'll still get there, and it is reversible. Okay, I'm going to stop there for now. Looks like we're going to have a part 3. So stay safe, guys.